It's a great pleasure to be here. First of all, thanks to all the organizers for inviting me. And I'm a bit nervous to be giving the first talk of this workshop for several reasons. As I was telling some of you, I haven't slept the entire night. I have a massive jet lag. So I need to make sure I'm awake the entire talk. And I have to make sure all of you are awake too. And hopefully exciting. Okay. So with that short note, let me introduce my lab members, a whole bunch of very enthusiastic students who come from biology, biotechnology, physics, mathematics, computer science majors, are interested in applying those techniques in the context of ecology and collective animal motion. And broadly in our lab, we are very fascinated by spatial patterns in ecology and the dynamics of these patterns. We study all the way from collective cell migration to landscape level patterns, such as the ones that you're seeing on the right side, vegetation patterns in semi-arid ecosystems. We try to come up with models that are not only explanatory, but also potentially of predictive value. And we don't always succeed. It's a really challenging stochastic non-equilibrium physics problem with the variations that biology introduces. It's a really challenging problem. We are interested in understanding the mechanisms of these patterns, functions of these patterns. And I started off as a physicist doing mostly theory and simulations. And thanks to a wonderful few years I spent in Ian's lab as a postdoc, I saw how experiments are done, and once I started my own lab, slowly we began doing experiments as well. We work with field data. So we sort of have a range of approaches that we take to address interesting questions in ecology. Okay, so let me begin with this. Because I am the first speaker, I think I took the liberty to put this really fascinating video of animal groups responding to predatory threats, and the presence of this highly synchronous motion and coordinated patterns that these organisms exhibit across scales is not only fascinating for an amateur scientist, but for scientists from nearly every discipline. And this is and what really makes to me very fascinating is that we can actually understand these in a quantitative way using computational and mathematical tools. What you are seeing here in the middle is simulations by Ian that showed that you can explain the kind of patterns we see in nature using simple computer simulations. So, and I was doing my PhD working on applying the ideas of phase transitions to ecosystems. And then I saw some of the works of Ian, and then that really got me inspired to sort of begin working on collective animal motion. So today I have two themes that I'm going to present. And in the first theme, I'm going to present some recent work that we have been doing on collective escape dynamics in animal groups. So you saw those in the previous video. There were these groups of animals responding to various threats, right? So now how do we begin to understand these large-scale phenomena happening in the nature, but in controlled conditions, so that we get a much better handle. But while this is a snapshot from an experiment where what we do is one fish has been trained to associate green light with a threat. So therefore it begins to move away the moment the light is switched on, and the entire group now moves. The information that only one individual had was transferred to the entire group. In the second example is that of a shepherd dog chasing sheep herds to specific targets, and how exactly does the information from the dog flow to the entire flock is the question that we are interested. Now we have gone from experimental setup to a field setup, but controlled experiments in the field. And finally, so these are black buck, which are an antelope species found in India. So what we have done is we have done drone recordings of these antelopes, and then we introduce perturbation to these herds. And again we look at how these herds escape from threats. And for today's talk, I am going to mostly going to present this part of this three story. These two stories are still developing. We are analyzing the data with the broad goal of understanding what are the general principles of information flow when animal groups escape from various type of perturbations and threats. So for today, I am going to present a paper which is going to come out very soon. This work was done in collaboration with Guy Théralos. So before I go to the presenting that in more detail, let me also tell you a second theme which I will present. So you see here groups of fish which are swimming synchronously. They're all in the experimental conditions. They all show high degree of coordination and synchronous motion. So the question we are interested in, what are the general features that we can extract from these data and present them in a to understand the stochastic dynamics that underlies the synchrony of the motion of these schools. Okay, so these are the two broad themes which I will present. So let's go to the theme number 1, understanding collective escape dynamics. So here you are seeing a shepherd dog chases a sheep flock. This dog actually follows the orders of a shepherd, and so the goal of this work was to understand, quantify the collective response of the flock to the herding dog, and how does the information transfer across the entire flock. Okay, so this is the setup. This is the shepherd who has this trained dog which chases the sheep based on the commands issued by him. I don't exactly know how these guys talk to each other, so don't ask me questions on those. My work, rather our work, starts once these guys begin to chase. So what the Guy Théralos, our collaborator in France, what they did was to put tags on each of the sheep as well as the dog as well as the shepherd, and then they tracked the movement of these, all the individual 14 plus 1 plus 1, 16 individuals at every 0.05 seconds, if I am right, over a period of a few hours. So if you now look, if you plot those trajectories, this is how it looks like. Okay, so you have all the sheep here at some point in time. This is the barycenter of the centroid of the group, and this is the dog. The dog is chasing, moving in a zigzag fashion. In response, the group is moving away. Okay, so let us first look at what are the basic properties of these sheep flocks. The first thing we find is that the sheep flocks actually have two sort of a bimodal state, it's a bistable system. So one state is a passive state where they have a low speed and low polarization. Polarization measures how synchronous they are when they are moving, and they also have a second phase which is a higher speed and a highly polarized phase where they are all synchronously moving together. And we are mostly interested in this phase because this is the phase that corresponds to response of the sheep to the dog. Okay, so if you look at the time series of various group properties, you will find that in this phase they are also highly cohesive. So typical body length of a sheep is around 1.2 meters, and you see that the cohesion of the group roughly is around 1 meter, meaning the near neighbor distances are roughly around 1 meter or even less. And this is also a highly polarized flock. So here we plot the polarization, group polarization, which is basically the summation of all the velocity vectors divided by the total number of individuals, and we take the average, the modulus. So this is like the magnetic order parameter plotted as a function of time. You see it is highly stochastic, but by and large it's in a highly polarized state, meaning the sheep are moving in the same direction for most of the time. Okay, so now these are the basic properties of the group. How does the information propagate in this sheep? So to do that, what we do is we compute directional correlations. What this basically means is that we take the time series of the two velocity vectors of different individuals pairwise. We take individuals and then we compute the cross correlation between them. And this cross correlation tells us which individual is following which. If the cross correlation was 0, they were influencing each other in the same way, so there is no leader or a follower. Whereas if this cross correlation was maximum, at a value, at a time lag, it would suggest that one individual was leading the other. Okay, so we compute this for all the pairs, and we also compute the strength of correlations, and what we find is a network of interactions between all the individuals. So here you see that the dog is at one edge at one end of the network, and all the sheep are here. And what we find interestingly, and if you zoom it out, what you find interestingly is that the individual who is at the front has maximum in degree. So maximum number of individuals are following the one at the front, and including the dog, whereas this number of degrees that are number of edges that is connecting to an edge reduces when you go back in the position of the herd.
So, Vishwesh, could you tell us again how you got the edges from the data?
So edges are the strength of correlations, whereas and for example, if I'm following you, the edge will go from me to you. Whereas if you are following me, it will be the other way around. Okay, and the size of the edge, so the size of the node, tells you how many individuals are following me. Okay, and the arrow represents the strength of the correlation.
Okay. What does it mean to follow? Yeah, that's exactly what I'm trying to understand.
So let's say if you turn after a few seconds, that means you are following me.
No, but I mean in terms of correlation, what does it mean?
In terms of, so basically, if you compute the cross correlation between our velocity vectors, if you find that the lag of, if you find the maximum value of this cross correlation, it will not be at 0. It will be at a finite lag, because you followed my direction after a little bit of lag. So every time I find a lag, that means that somebody is following the other, right? Yes.
So presumably you have, when you calculate those cross correlations, especially with the lag, then you get non-zero correlations for basically every pair of nodes.
Absolutely, yeah.
So you're applying some
use a
sort of
threshold.
thresholding.
Yes.
So with the results that you're seeing, so first of all, was there, how did you go about picking the threshold? And then did you test whether the results that you're seeing are robust to when you kind of play around with it?
That's a great question. We do use a threshold, which is around 0.5 to 0.8. So the number of edges you find will reduce if you have a higher threshold, but the overall structure doesn't change. So what do I mean by overall structure is this graph really. So here I am plotting the in degree. So 0 means this individual has nobody who is following, whereas this individual has 12 other individuals who is following. Okay. Now on the y-axis, I am plotting the relative position along from the centroid of the group. A positive number here would mean that I am at the front of the group, whereas a negative number would mean I am at the back of the group. Okay. So what I mean by the structure does not change depending on the threshold is that the fact that there is a correlation between the position in the group and the in degree, this does not change depending on the threshold I choose. But the number of edges I find, those will change. Okay. And so there are basically two interesting findings that we do here. There is a spatial fidelity. Some sheep are typically in front than in the rear. So some sheep seem to be typically at the front, and we were expecting that because this is a dog that is chasing the sheep herd, so the information flow must be from the back to the front. But surprisingly, we find that despite that, the guy who is at the front, right, so the positive number would mean somebody is at the front. The frontal guy has maximum number of followers. That means that the information is actually flowing from the front of the group to the back of the group, despite the fact that it is the dog that is actually chasing the entire sheep and trying to move the sheep to a target. There is actually a target in the entire ground where the dog is actually trying to move. So what this really tells is actually the following. On short time scales, which this network is based on computing the correlations over really short time scales. When I say short time scales, within a matter of few seconds, on those time scales, the collective behavior is driven by the local interaction among individuals and typically driven by the one who is at the front. The frontal individual decides where to go, not the dog. Dog decides over long time scales where the entire herd must be, but over the short time scales, the sheep at the front decides where to actually go. This was an unexpected result, and this could potentially be, yes?
Yes, quick question. Can you tell whether the follower is following the leader or following a follower of the leader?
There is a network, yes, absolutely, yes. So if you go back here, so this is a follower. So this follower is following many other individuals. So this is the sort of the leader, right? This guy at the front, right? But this guy is following 3, 3 is following 14, 14 is following 6. So it is a nested network. It is just that the guys at the front have more followers than the guys at the back. Okay, so 4 follows 3, 3 follows 14, 4 also follows 6, 6. So it is a very dense network. And what we find is that if you increase the threshold, some of these edges will drop off. Okay, but the overall structure would still remain. Yes, there are 2 questions there. Yes.
So do they switch leader, or is this this is just for some short time frame, or is that structure?
This is an average over the entire.
And if you look at it over time, does this network change a lot?
No, it doesn't, because this is basically an average over the entire thing. So for example, 6 is typically at the front, whereas 11 and 5 are typically at the back of the herd.
But if you look at
very short time scales,
yeah.
yes. There is of course variability. Yeah, but the variability, you will find that it's hard to distinguish where these central guys are. So they sort of shuffle the positions a lot, whereas the one at the back edge and the one at the front, they are relatively more stable in the entire herd.
Would that be a property of motion in space, or would that suggest that it's more of a star network than, that all the ones in the middle are following the front, and therefore they're not ordered in their minds relative to each other? Or is that just a property of motion in space?
So this was also a question we had. So what we did was we did a null model. So we had a null model where we actually built a herding model. So can these results be reproduced by a model? Because then we began to understand what's really happening in this data much better, right? So what we did was, so I will not go into the details of the model. So it's like a typical collective behavior model with a new rule. Okay, that typically in a collective behavior model, there are individuals, they are following their neighbors, they do an average of the orientation of the neighbors, they attract towards the neighbors. In addition, we now have a dog, right? This dog has 2 properties. It has a property, it wants to move the entire herd towards this target. That is one behavior it has, and it does it whenever the group is cohesive, it tries to move the entire group towards the target. On the other hand, if the group is very spread out, it first tries to make them cohesive by making sure this guy comes here. So we assume that the sheep just repel the dog. That's the simplest response that the sheep has to the dog. But dog has these 2 behavioral rules, and typically the rules of collective behavior, or the standard rules of collective behavior here, nothing new that we have added here, apart from the fact that there is a repulsion from the dog. So now the question is, if the observed behavior in the data was a general enough pattern, this model should also be able to reproduce that, right? So in fact, so this is how a model simulation would look like. So this is the dog which is chasing the a flock of 14 dots, okay, in this case, mimicking sheep. So now if we construct the similar network, okay, so basically now I have the trajectories of the model individuals, not the real sheep or real dog, and if I apply the same network analysis, starting with the time series of the orientation of all individuals, calculate the cross correlation, find if the correlation is significant, find if there is a leader follower bit for each pair, and construct the entire network, and you find that the model reproduces the patterns of data pretty well. We also did one more thing which I don't show here unfortunately, which is that if there was no dog, if there was a spontaneously moving herd, would you find a structure like this? It turns out that we do not find a structured network at all. Okay, we do not find that basically the key idea here is that some individuals are always leading, whereas some individuals are always following. That structure just dissipates the moment we sort of do simulations with no dog. Okay, I have 2 questions. Yes. Yes, you want to go first? Yeah.
I'm having a little bit of trouble here in terms of the way that information flow is being discussed, which is that, like, the assumption is that the output of the information flow is alignment, right? That we all kind of match on one thing, but the leader is not making value neutral movements, right? It's moving because individuals are kind of pushing behind it to kind of make its. Is this in here?
Yeah. So I mean, I've had to really break my head about this, So, but in this context, direction seems like a correct information to me, because at the end of the day, the dog are trying to, put this herd into the target spot. So that's a very directional movement. So I think there is no ambiguity that it is direction, and we also conducted a similar analysis with the speed of individuals. So speed also varies, right? They're not moving at a constant speed. We find the exact same network, but the strength of the network is much weaker. In fact, much weaker. This is a really, really good, very strong, signature of strength here. But whereas with the speed, it's much weaker, significant but much weaker. But therefore, which suggests that the speed, direction is actually I think a right metric of information here. Yes.
In your model, are your sheep identical?
Yes. In the model, they're identical.
But how could it be that one emerges stably as the leader?
So usually what happens is in a simulation, it's a really, really great question, okay. So what really happens is in a simulation that is run for a comparable time unit as the real data. So the one who's at the front typically remains at the front in the simulations as well. So in fact, in the real data, the frontal sheep, as I mentioned, there is a spatial fidelity. There are some individuals who are always likely to be at the front. We do not encode that in the model, but that does happen spontaneously for short runs of simulations in the model as well.
So I'm interested in this question about the naturalness of forward motion. Have you coded it up with sheep in the back with eyes in the back of their head?
So we do assume that they cannot see some cone at the back. Yes.
So to what extent do you think your results are artifactual of having eyes in front of your head?
Why would you call it an artifact though? That's a real phenomenon. I don't know why would that be an artifact. It's an interesting thought experiment to do if there was no sort of visual cone that sort of if it could see everyone around, would the results change? Okay, maybe my student may have actually done this. I don't know. Yeah.
Contributing to that discussion. So when you made the statement, I think the sort of controversial statement that we're discussing here is the flow of information, and that it flows from the leader of the herd back to the dog, where we would think intuitively it's the other way around.
Yes.
So a little gripe that I have with that statement is that it's based on correlation. So you're testing for linear relationships. And I think maybe what can contribute to consolidating the picture here is that you said that this is on a short time scale, which to me means when you take nonlinear dynamics and linearize them, that's the time scale that you're working on, right? So in the nonlinear relationships between the dogs and the sheep, the sheep and the dog, might very well indicate that the dog is actually leading the movement, and I think that is what you may have hinted at by saying that on a longer time scale, you do see the dog having a higher influence, but you don't see that on the shorter time scale. Yes. So maybe that feeds into that discussion.
So to be very honest, as I told you, I showed this. This is a paper which has more details, all the technical details. So to be very honest, I do not fully understand. Let me be very honest. So we have these two work which are doing something very similar but using different setups. So and we are doing similar analysis, including this fish experiments, right? And again, we are constructing. This is a really well-controlled experiment. So this has a very different dynamics, right? So in this experiment, there is a fish that really leads, and we know who is the one because we have trained them, And then we also have controls where nobody is trained. There is no really an individual with any information. So we are doing exactly similar analysis to sort of integrate all the three work to make a better story and to understand can I make a more cohesive, strong statement, which is why all this feedback is really, really helpful. So these two work are still sort of ongoing. Maybe if I finish all of them, I could answer you. So what we're also trying to do is think of a very general model framework that has these two type of contrasting escape responses. One in which somebody pushes you from behind, like the dog chasing the sheep flock. The other one is where somebody gets an information either about a food source or a migratory direction or a threat in some direction, therefore moves away from the group and everybody else follows. So that's like a pull. The entire group is being pulled. So how do these network of information differ between these two scenarios? What would model tell us and what do these data sets will tell us? That's a story that is still being done, so I don't have all the answers, but I do think broadly speaking, we know for a fact that the dog is pushing them to the target on a longer time scale. It's a short time movement adjustment which is very surprising. It is still driven by the sheep. I think that we are now very confident to be able to say that. I have what's the time now? I have 20 more minutes, right? Okay. Okay. So can I go on? Is it okay? Because I have a theme 2 which is even longer than what I have so far.
I just had a very short question.
Yes.
So the dog obviously is different, and in the sense that the way that sheep react to its movements is different than sheep react to sheep movement.
Yes.
So in when you construct this correlation matrix, the fact that it's kind of the last in your network may not be so meaningful, right? Because it just means that its motion is not as correlated as the motion with the other things, but it might be moving in a different way to influence them. Do you see what I mean?
But the strength of correlation is pretty high. I wouldn't say that the correlation is not strong. Where is it the dog positioned in the network is different. The strength is very large. In all these cases, we find the strength of correlation to be of the order of 0.8 to 0.9. So yeah, it's pretty high. So I wouldn't worry about the strength part. Where is it positioned is the more interesting.
I'm just saying it's a different correlation, so the fact that it's different in the network is clear, but interpretation is not so.
Yeah, sure.
But Dima, it's also, you reduce this to a one-dimensional problem where you're constantly monitoring the average velocity of the flock, but the dog is doing something in 2 dimensions, presumably, right? So
We are looking at the 2D velocities of every individual here, right? Every individual.
Well, you've reduced it to a number, so it must be the relationship between the average velocity vectors.
No, because this network, it's basically for example 8, this individual, I will do a pairwise correlation for everybody else, and that is done for everyone in the entire flock, including the dog. So it's everyone is treated equally when constructing the network, and it is just that the dog appears here. And
No, I guess I'm not clear. Maybe I'm misunderstanding, I'm sorry. So what your correlation function is about the relative velocity according to some average for the whole flock, right? And that average is changing.
It's between two, it's pairwise velocity.
But I mean, well, same difference, right? But it's still, you're taking a magnitude relative to some average.
Yeah, maybe yes.
So you're basically on a manifold that's constantly.
Yeah, basically we are always measuring everything, you're right, to the relative to the instantaneous group velocity. Yeah. But the calculation isn't exactly identically though. Dog does move, chase that, sort of a oscillatory pattern around the group centroid, which I showed in the beginning. Maybe we can discuss this more. Thanks for all the wonderful questions, thought-provoking and interesting. So, okay, so let's hope that when we put all these together, we get a nice, interesting story that explains all the puzzles. Okay, so now let me move on to the theme number 2 of my.
I know you really want to move on. you made this great prediction from your model that if you remove the dog, then this goes away, right?
Yes, absolutely, yeah.
So just track sheep without the dog. I bet you it doesn't go away. Because there's probably all sorts of hierarchies going on, but that's a testable prediction.
Yeah.
Did you test it?
If the thing is, what happens in the real sheep is that the moment dog goes away, they also don't polarize. They just stand still in the real world. They just stop eating grass. So yeah, it would have been really great if yes.
It looks like the dog keeps fairly large distance away from the flock, and in your simulation.
The other way around, the sheep really move fast to keep the distance.
Well, that's fine.
I think both are correct. Actually, dogs are actually trained not to really harm these.
No, the question is not harming. The distance allows the leading sheep see the dog, because if it were pushing it from behind, that would be different. And in your simulation, the dog is very close.
I can always parameterize that number, so it's not a really. I can always sort of parameterize the repulsion zone for the dog, so that's an easy parameter to play around with. Okay, so okay, sorry. So here I'm showing you. Oh, sorry. A bunch of videos that I've stopped. Okay, of various species of fish. So the question we are now asking is: are there universal dynamical patterns of fish schooling? Okay, they are, they're all different species. They all look a little different, but they also all highly synchronized and school. So what can we say about them? So the key point that I would like to make in this context is that the stochasticity of finite flocks is really, really important. So what do I mean by that? So when we think of order parameters in physics, we are often interested in looking at the macroscopic behavior, right? However, the order parameter for these 15 individuals is going to be really, really stochastic. And in fact, this is a realistic scenario. Many flocks are small. Of course, there are really, really large flocks as well, but many organisms do show small groups. All the experiments are often done in a small experimental setup, right? And in these small scales, the stochasticity cannot be really ignored. I will come to that, what I really mean, in a short time. And they can take non-trivial form and produce some non-trivial effects. So I will really try to explain what do I mean by this, both theoretically as well as using data. So what we really need is a theory of finite flocks. And in particular, I am interested in this quantity of group polarization, which is the analog of magnetization for the fish schools. Okay, so what I will do is in this part of my talk, I will present a theory of group polarization accounting for the fact that the number of organisms are finite, and I will show some experiments, the same experiments, and we use the technique of data-driven modeling to see if the theory that we build, sort of comes back with these data-driven models. Okay, and then following this, we have done some interesting work. If I have time, I will briefly touch upon them, or otherwise I will skip those. Okay, so what do? We compute this group polarization, which is the average of the velocity vectors, orientation vectors. I am ignoring the speed here. So if I plot the time series, this is how the magnetization order parameter looks like. You can see it's pretty highly synchronous for a large time. It's also highly stochastic. So my point is you should not ignore this stochasticity. Okay. So typically we have taken the approach of individual based models which sort of encode rules for each agents, such as follow the near neighbors in terms of direction, in terms of being attracted towards them, and then one adds a stochasticity on top of that. That's a sort of classic ways of modeling collective motion. On the other hand, if you look at the physics coarse-grained approaches, you define the flocking order parameter, and then you write down hydrodynamic equations such as this. This is a very sort of toyish version of the Toner 2 model, which has a deterministic or a drift term, which is cubic, which has a spatial coupling terms between the order parameter and the concentration, and then you have typically extrinsic noise term, which is a thermodynamic noise term, and typically these are all written for the macroscopic scales, right? Now, so this equation really cannot be used for describing the small sized flocks, and we really need a different theoretical framework. So the idea is that the spatial coupling doesn't really make sense anymore because it's really one blob. of 15 to 20 individuals. So we should really think of the rate of change of order parameter as driven by a drift term and a diffusion term with a Gaussian white noise. Okay. So what are these drift and diffusion terms? So let us see, can we build a simple spin model where we assume some fairly simple rules. We assume that an individual copies a randomly chosen individual at a rate c. In addition, it also does a stochastic turning without copying anybody else. Okay, this is an extremely simple model. For these two sets of rules, what would be the dynamics of group polarization? So what we do is, we can actually, I won't go through the entire sort of mathematical formalism here. So the idea is to write down the Fokker-Planck equation and do a Fourier expansion of the step operators. We can do that. When we do that, the crucial point here is that because this n is now not very large, I can't drop all the terms. So I have to keep some terms which have an explicit n dependence. If I do that and use Ito's lemma, we can actually derive a stochastic differential equation that tells me how the order parameter changes with time. Okay, let us understand this equation. Okay, so this equation has two terms. So let me ignore the stochastic term first. If I look at only the deterministic term, it shows that the order parameter basically exponentially decays. Okay, which means that this simple pairwise alignment, an individual copies a randomly chosen individual, if that is the only rule that the fish are following, there should be no order if this term has no effect, or if this term has very minimal effect. Okay, so here is the key interesting insight. This insight that if we have a potential of this type with a stable equilibrium in the middle with some noise will give rise to a steady state distribution around the stable equilibrium. This intuition is correct only for additive noise, when the strength of the noise is constant. Okay, that approximation would be true only when n is really large, for large group size limits. However, the moment n is really large, when n is small, like 15 individuals in the case of my experiments, this stochasticity actually has a non-trivial form. So again, pay attention to the form here. A typical thermodynamic noise will not have the m square term here. It would be an additive noise which is a constant like this. But now we have a m square dependence. What this really means is that when the system is in the disordered state, the noise is highest. When the system is in the ordered state, the noise is lowest. So what this really means is that as the ball is trying to settle down to the equilibrium, the noise will push back, and that can lead to a bistable system, even though deterministic dynamics did not really have those effects at all. Okay, and basically what I am trying to show here are two possible outcomes of the simple pairwise interaction model of fish schooling. There are two possible outcomes. Do we really find them in the real data? I don't know, let's see. Now that is one possible model, but it's an extremely simple rule, right? Copying one individual randomly. But typically, if you think of classic models of flocking, you do an average interaction. You look at your neighbors, you do an averaging, and then you move in the direction of average. If you do the same Fokker-Planck analysis, you will find a different stochastic differential equation. I will contrast these two in the next slide. So let us look at the deterministic or the drift terms here. You can see this now has a cubic term like the Toner 2 formalism. Okay, so I have plotted here the drift term for the pairwise interaction model and the drift term for the ternary interaction model. The fundamental difference is that in the pairwise interaction model, disorder, order parameter 0 is stable, whereas in the ternary interaction model, disorder is unstable, whereas the highly ordered state is actually stable. Okay, that's the fundamental difference between these two models, and the diffusion term has a very similar shape. The shape of the diffusion term doesn't really change, but the fundamental difference is that the ternary model admits a deterministic ordered solution, whereas the pairwise interaction model doesn't have a deterministic ordered solution. the order in the pairwise interaction model comes only when the group size is really small. Okay, this is all nice theory, and one way to think about this in a pictorial way is using these potentials. The ternary interaction model is like a bistable potential. Sorry. Actually, one way. So disorder is unstable, order is stable. You see these 2 times primarily because m and minus m are same in the sort of in the sense of order parameter. The vector m and vector minus m are basically same, right? So that's the reason why you have it's a spurious bistability here. Truly is only one stable state. Okay. So and what we did was we also done this. So all that I showed you was a non-spatial model. We implemented the same rules in a spatial setting, like a Vishwesha model, and we find the exact same results. So these principles are true even in a spatial setting. Yes.
Wouldn't it be effectively the same if the time scale was different? So if you're randomly choosing neighbors,
that's a great question.
So in a very fast time scale, it should be the same as averaging.
Yeah, but it's not.
It's not.
It's not. Yeah. It's really it is not. Yeah, they are fundamentally different. Yes.
Even if you separate the time scales.
Yeah. I think so. I think that's the most. That was precisely, in a sense, very surprising to me as well. Yeah. Okay. So now, are these fish doing anything as simple as those equations? All those equations are fairly simple, right? We didn't really have many complexities of what real fish do. Real fish do amazingly complex cognitive tasks. When you say 10 minutes, it includes the discussion time. Okay, excluding the discussion time. Okay, great. So, but do these fish really follow those really simple stochastic differential equations? Okay. Now that's a really hard question to understand, right? Now I have here stochastic differential equations and I have time series data. Data is time series, but the theory is stochastic differential equation. So how do we now map them? So it turns out that, again, let's now look at the typical time series of 3 different species. They all look similar. The gaps you see here is because of the tracking issues. Okay, and if you look at the histograms of the order parameter, they all look very similar. Okay, this is the 2D histogram, this is the 1D histogram. So mod m close to 1 would be in highly ordered state. Okay, so they all, that means maybe they're all same, they're all doing very similar things. Okay, so now here we use an idea called data-driven equation discovery. So the idea is that from data can we derive the dynamical equations. Okay, so I will not go into too many details. It turns out that it is indeed possible. If I give you a time series data, I can actually sort of discover it is possible to decipher the underlying dynamical equations that generated the time series, the generated time series. And it's actually a really powerful method. The basic idea is, for those who are familiar with the physics, is to compute what are called the Kramer-Moyal coefficients from the time series data. And the first moment basically corresponds to the drift. The second moment basically corresponds to diffusion. Okay, and once you have constructed them, you now do a simple any of your favorite statistical technique to find underlying functions, and those would actually then give you the f and g functions. Okay, so in fact, I wish I had that slide where we had identical time series that we simulated from very different equations, and we have developed a package in Python. When I say we, it's really my students who know Python. I am an ancient guy who knows C only. Okay. And this Python package takes the time series as an input and spits out equations that does the best job of describing the data. Okay. So the preprint and the packages are all available for anybody to check out. I think it does a really beautiful job. Okay. So we applied this method to this fish data set. Jitesh was the one who led this work. We found that these fish follow this stochastic differential equation. Okay, I will show you a next slide where I have the data plotted. You will see an amazing similarity of the terms. The deterministic term is a linear term, exactly like the pairwise interaction model. The diffusion term has this 1 over mod m square feature, which is also exactly the diffusion form which we had for the both the models, right? And so basically what we infer from this is that these fish show highly ordered schooling behavior, but the deterministic model does not predict order. So the only way order can come about is because of the stochastic term. So we call this noise-induced schooling because the stochasticity in the system, which is inherent to the inherent because of the small group sizes. Actually, I'm going to skip this. Sorry, really sorry. This was to compare one of Ian's old work on locusts. Locusts and these fish are different. So the drift here is a plane, whereas this is a cubic term with order being unstable. This is how these two systems are very different, although diffusion terms are very, very comparable. Okay, so now let's ask, what happens to other fish? We have so many fish. They all had nearly identical histograms or PDFs. In physics, we often look at correlation functions of time series and the PDFs. So what we show is that even when you have identical PDFs and comparable correlation functions, the underlying equations can be really, really different. And in fact, I will now have to rush. So what we did was, for the rest of the work, we sort of also accounted for the fact that fish have a variable speed. So we constructed not only an equation for m, but an equation for both the order parameter as well as the speed, a coupled differential equation that captures both the synchronization as well as the speed of the group. And in the 2D, what we now have is we have constructed the null clines, okay, and the fixed points from the data. So this is as good as constructing an equation for the underlying model, right? So because whenever you have a model of coupled differential equations of two variables, what we do is we plot the null clines and the fixed points, and then we look at the entire flow diagram to look at where are the equilibrium points. So now what we find is that there are two types of systems here. One system which has only one fixed point at the low order. The other four fish have bistability. So the blue dots here represent stable equilibrium, and the sort of these open circles represent unstable equilibrium. So the four fish have this bistable dynamics. Now what is really interesting is that if you looked at the videos, if you looked at the time series, if you looked at the histograms, they all looked very, very similar, but the underlying dynamics are actually fundamentally different. But fundamentally different, yet they have broad classes. Okay, and let me now sort of describe the three classes. The class 1 is a noise-induced order, because in this system, the deterministic stable equilibrium is at 0 order, but the observed data has a high order. Now, how did the system? Why is the system not showing a peak here? That's because the stochasticity is very, very high in this state. It pushes the system to high order. So therefore, we call this noise-induced order. In the second class of system, which 4 of these species belong to, there are 2 stable equilibrium, but again the mode is mostly at the high order. Okay, so again, if you look at only histograms, this histogram and this, they are nearly identical, but the underlying equations are really, really different, bistable system. But again, we don't see a mode near here because the stochasticity here, pushes the system away from the disorder to the higher order state. And we also predict there must be a third class called deterministic order, like the Toner 2 model, which is probably what the locusts seem to follow. Okay, and but we did not find any of our fish species following this, this equation. Okay, so I have 1 minute. So yes, the answer to are the general universal dynamical behaviors of the fish schooling, I think yes, despite the fact that there are very different species showing synchronous schooling, I'm pretty sure the exact rules they follow might be really different in terms of what they actually do, but at the group level, they seem to have few classes such as deterministic order, noise-induced order, and bistability. Okay, so this is to sort of place this in the framework of Toner 2's work. The main contrast being that the focus there was on the macroscopic dynamics, whereas we have focused on the small group size mesoscopic dynamics. Okay, so I will sort of jump and sort of.
Can I ask a very quick question?
Yes, yes. Now I have time for discussion, so I'm sort of done. I have only a few slides which I can ignore. Okay, I can I can ignore synthesis. Okay, I'll stop here. So I'm happy to take questions and discuss for the rest of the time.
Thank you.